I'm Sarah Traynor, research scientist at Active Motif, and in this video, I'll guide you step by step through formaldehyde fixation of cells in a 96-well plate, an essential preparation step for your TIP-ChIP experiments. TIP-ChIP, or Tegmented Indexed and Pooled ChIP, is a low-input multi-sample ChIP assay that enables the processing of up to 96 samples per target in a single IP. This approach reduces sample-to-sample variability and delivers greater consistency across experiments. It works with as few as 300,000 to 1 million cells per sample, reduces technical variability between samples, and is ideal for large cohorts, time course studies, or drug treatment experiments. TIP-ChIP is a plate-based assay where each well is a sample. The samples are fixed in a 96-well plate. This will be demonstrated within this video. During the assay, a unique tegmentation event occurs in each well that barcodes each sample and enables pooling. An antibody of your choice can then be used to profile all of your samples at once. Now diving into the steps of successfully fixing your cells for TIP-ChIP. First, collect your cells. If you are working with adherent cells, detach them using EE or trypsin. The remaining steps will take place in solution. Next, count and aliquot your cells. Each well of a 96-well plate will be a unique sample and requires 300,000 to 1 million cells. We recommend using a Corning 96-well plate. The cells will be fixed within the plate. The fixation buffer will be added directly to each well of the 96-well plate containing the cells and the culture media. After 10 minutes, fixation will be stopped and the cells will be washed twice with cold PBS. Throughout the process, a pellet should be visible after every spin step. Lastly, the cells will be snap frozen in liquid nitrogen and stored at -80 degrees Celsius. Accurate cell counting is critical because after fixation, you won't have another opportunity to recount your cells. If you're working with adherent cells, first detach your cells using EE or trypsin. Once you have a single cell suspension, follow the same procedure as you would for suspension cells. Each well should contain 300,000 to 1 million cells. Adjust the volume so each well has exactly 100 microliters of the desired cell number to streamline fixation with a multi-channel pipette. Remember, always use a multi-channel pipette to improve consistency and efficiency. Ensure you have viable cells prior to starting the experiment. Ideally, cells are over 80% viable. Here we have an example of a counting field that shows a single cell suspension with high viability. Begin by ensuring your workspace is clean and all materials are ready. Make sure to have pipettes, timer, ice, and template sealing foil from USA Scientific. Prepare a 10x fixation buffer using our recipe available on our website and add formaldehyde freshly right before use. It's best practice to add formaldehyde after cells have been aliquoted into the 96-well plate. Ensure your swing bucket centrifuge is chilled to 4 degrees Celsius. Stop buffer for stopping fixation can be aliquoted into PCR strip tubes for easier addition with a multi-channel pipette. Keep 1x PBS on ice and reservoirs ready for wash steps. This demonstration is going to be done with one column of a 96-well plate or 8 TIP-ChIP samples. Quickly add the previously prepared 10x fixation buffer containing freshly added formaldehyde to each well using a multi-channel pipette, working swiftly to maintain consistency. After adding formaldehyde, place the plate on a nutator at room temperature for 10 minutes. After 10 minutes, remove the plate from the nutator. Stop fixation by adding stop buffer at a 1:20 volume ratio using a multi-channel pipette. Chill the plate on ice for 5 minutes, then centrifuge at 800 G for 5 minutes at 4 degrees Celsius. A good visual QC is to look at the bottom of the plate after the spin to ensure there is a cell pellet. This can be done all throughout the TIP-ChIP protocol after each spin step. After retrieving your plate from the centrifuge, quickly and decisively invert the plate to remove the supernatant in one swift motion. This method ensures consistency. On ice, add 100 microliters of cold 1x PBS to each well without resuspending the pellet. Incubate the plate on ice for 2 minutes. After the incubation, centrifuge at 800 G for 5 minutes at 4 degrees Celsius. Once again, quickly and decisively invert the plate to remove the supernatant in one swift motion. Repeat this wash step once more for a total of 2 washes. During the second spin, prepare for flash freezing by retrieving liquid nitrogen. After the second wash step, remove the supernatant once more, leaving behind the fixed cell pellet. Properly dispose of the supernatant. Seal the plate using the USA Scientific temp foil, ensuring that all edges are properly sealed. Snap freeze the cells by placing the plate into a bucket with liquid nitrogen. Remember, accurate cell counting, precise fixation timing, and thorough supernatant removal are crucial. Regularly check that a visible cell pellet remains throughout the protocol. For any additional questions, visit our website for the complete written protocol or contact our tech service department. You are now ready to successfully perform your first TIP-ChIP experiment.